Hi everyone, Mrs. Below here. In this video, I'm going to be talking about the dark reactions, also known as the Calvin cycle, and also known as the light-dependent reactions. So previously, we talked about the light reactions or the light-dependent reactions. Those reactions depend on sunlight because you need that to energize the electrons, which came from the splitting of the water. Those electrons actively pump that H plus that came from the water across the membrane. So chemiosmosis can occur through that ATP synthase to make ATP. And then those electrons get re-energized at photosystem 1 in order to make NADPH. So now we're going to use both those things we made in the light reactions here in the dark reactions. Now, as the name suggests, it doesn't happen in the dark. It just isn't using sunlight. Because think about it. If you don't have sunlight, this won't occur. And then therefore, this would not occur. So you can also call this the Calvin cycle or the light independent reactions just because these reactions aren't using sunlight. But we're going to use the two things from the previous reactions, ATP and NADPH, and also that carbon dioxide that plants take in. And ultimately, we're going to make glucose and oxygen, which actually, hold on, I have to please fix this in your notes. I realize I still haven't fixed this. Oxygen should be over here because that's where we're making the oxygen is once we split up the water. So please make that adjustment in your notes. Okay, so let's go to the dark reactions. So the light reactions were occurring kind of inside the thylakoid and on the thylakoid membrane, those little pancake-like things. Now we're going to be in the stroma, which in the chloroplast is just the stuff that fills the chloroplast itself. So the reactants in this reaction are the products ATP and NADPH, which we got from the light reactions. And this is happening in the stroma, which is, again, like I said, the stuff that fills the chloroplast. Versus light reactions happened inside of the thylakoid / on the thylakoid membrane. So this is the Calvin cycle. It's very similar to the Krebs cycle. And we're going to break this down into 3 main steps. So you get this diagram on the test. I'm just going to be adding some stuff to this diagram. So we kind of have 1, 2, 3 phases that we're going to go through. So the first one is called carbon fixing. So we're going to use 3 CO2 molecules that enter through the stomata. Oh, I'm sorry, that enter the stroma. We haven't gotten to stomata yet. Cross that off. That's a typo. This should say inside the chloroplast. And because that's where these reactions are occurring. And since this is just like the Krebs cycle, we're actually going to go around it 2 times because we're going to use a total of 6 carbon dioxide molecules because of the balanced chemical equation, and this reaction is showing 3 going through. So we are going to go around this 2 times, just like the Krebs cycle. And we are going to use an enzyme, kind of like that coenzyme A. This one has a more fun name. It's called Rubisco. And it is going to basically combine that carbon dioxide with something called ribulose biphosphate. You could just abbreviate it RUBP. So basically step 1 is combining this molecule and carbon dioxide together using an enzyme, kind of similar to what you saw in pyruvic acid conversion with cellular respiration. and then it's going to make this molecule here. And then this molecule is actually made from the breakdown of the unstable molecule made by the combination of these two things. And that is the first phase. Phase 1, carbon fixing. We're taking carbon dioxide from the atmosphere, we're in the stroma, and we're combining it with a molecule called RUBP. Now we're going to see where those things come into play from the previous reactions. So now we're going to do a couple things to this molecule that we just made. And so it will be rearranged using ATP and NADPH. Sometimes you also might see this called like reduction or oxidation, but we're just because we're not really getting into that depth to the chemistry, we're just gonna say these things are rearranged. It's a chemical reaction. So ATP is going to give a phosphate. So instead of really, I guess, being used for energy in this case, it's really just being used as part of a chemical reaction. And that becomes ADP. That will go back to the light reactions to reform ATP. And then NADPH will, of course, drop off a hydrogen or electron, right? It's a hydrogen electron carrier, and it becomes NADP+. Those both go back to light reactions to be remade again. So that's why you need sunlight in order for this reaction to occur, because these two things were made from the light reactions. Now, I know in the diagram that this and this look the same. Chemically, they are two different molecules. So that was step 2. Now we're in step 3. So we're going to have one of those molecules is going to exit the cycle. So we kind of like set aside one of those molecules. There's 6 of them right now. One of them gets set aside. We're going to use that to make glucose. Because then the second turn, when we set another one aside, then we're able to make glucose, that's C6H12O6. The rest, those other 5 molecules, we're going to, so this one exits. The rest of the 5 molecules are going to continue. We're going to use ATP again. So the 5 remaining 3 carbon are made back into RUBP, because this is, of course, a cycle. So technically, RUBP is the thing that gets regenerated. Very much analogous to oxaloacetic acid that you saw in the Krebs cycle, so that we can continue this cycle again. So when all is said and done, we use 6 molecules of carbon dioxide, right? We went around twice through the Calvin cycle. And we also used ATP and NADPH, which were from the light reactions. And they become ADP, because ATP drops off a phosphate, and NADP+, which drops off a hydrogen. And those ultimately go back to the light reactions in order to be remade. So that's why we need light in order for this to occur. And after we go through the cycle twice, we set one molecule aside the first time, we set a second one aside, we combine those together to make one molecule of glucose, which is C6H12O6. We could possibly use it to make other organic molecules, depending on what the plant may need.